Hi everyone. So these notes are going to review the basic stuff you need to know about enzymes for anatomy. There is more to learn about enzymes beyond what I'm covering that you may have familiarity with if you do AP biology or bio honors, but this is just going to go over the bare minimum that you need for anatomy. Okay? So in these notes, we're going to review what enzymes are, some key features in enzymes, why dedenaturation is critical and what it means, as well as what environmental factors in your body can affect enzymes. And the reason why this is important is because enzymes are critical to your body, so we really need to understand how they work and how homeostasis is all about preserving those enzymes. In this lesson, you're also going to go over these terms. Okay? Now, you're also going to notice in this lesson the numbering is slightly off, and the reason why is originally this lesson was combined with the metabolism lecture. So just a heads up. So, and this is just a quick review of metabolism where we went over catabolism and anabolism and how one is about releasing energy and building up and breaking down molecules, and the other one is about building up and making bigger molecules and taking in energy. Okay? So, originally this was part one of the lecture, and then the enzymes were part two of the lecture, which is why the numbering's a little off. If this bothers you, you can change this number to one. Otherwise, don't worry about it. Just take this lesson on a whole separate notes regarding enzymes, and you're all good. So now that we know about reaction types, now we can go into where enzymes play into all of that. Now, here's the deal. Anabolic and catabolic reactions, both of those require energy to at least kickstart the process. Maybe overall, you'll get energy out of catabolism versus lose energy out of anabolism. But either way, both require some amount of energy to kickstart the reaction. Enzymes come into this because they are the things that allow the start of those building and breakdown reactions at a level that's doable by your body. Okay? So in this case, they're protein workers that are needed to help speed up your chemical reactions so they occur at a reasonable pace and under reasonable amounts of energy. Okay? So forming or breaking bonds require energy to start the reaction. So maybe you get energy overall, but you need to put a little bit of energy into it. Okay? Now, the amount of energy needed to kickstart it can be dangerous. For example, we burn down or break down sugar all the time to make ATP molecules. But outside of the body, without enzymes, the amount of heat needed to start the release of energy from sugar is basically the heat of a fire. Now, that's dangerous in your body. So, enzyme proteins lower the amount of energy needed to release to break down or build up molecules to safe levels. So in this case, without enzymes, bodies can't do enough of the reaction for your body to stay alive. If you don't have enzymes, you either need dangerous levels of energy that can damage your body, or those reactions that occur would occur at a very slow rate compared to what you would need. So instead of something taking minutes to do, you may take hours, weeks, or months to do the same reaction. So the processes that help you digest food that may take a few hours would take months to occur outside the body. Okay? So enzymes let reactions occur in a way that's sustainable for you to stay alive, whether allowing you to do it at an energy level that's safe or allowing you to do it at a speed that'll keep you alive. So as stated earlier, enzymes let those reactions you need for your life to happen under safer conditions. Okay? And if you lose those enzymes, if those enzymes aren't working, that's a problem. So generally speaking, when we talk about homeostasis, maintaining conditions within your body, a lot of the time, those conditions are important to allowing enzymes to still work. They're very sensitive to changes in your body. So homeostasis often is about keeping your enzymes functional. Okay? So just reminding you, energy, so this overall is a reaction that's going to release energy because your starting stuff called your substrate or reactants have way more energy than your products. This should be familiar to you if you took chemistry, whether it was regular or honors. But the amount of energy needed to kickstart this reaction is way higher and thus more dangerous or takes a lot longer than the amount of energy that needs to kickstart the reaction if you have an enzyme. Okay? Now, I'm not going to really test you on this graph, but hopefully it helps remind you why you want to reduce that energy level to make these reactions work better in your body. Okay, right? It's a lot less effort if you have to put in a little bit of energy to get the same results compared to having to put in a lot more energy. So enzymes mean that the amount of energy you need to put in initially to get the same results is a lot lower. Okay, so this is a classic way enzymes work. So the blue stuff is the enzyme. So its job is basically it grabs the reactant and helps let them do whatever you need to do. It can either take two separate molecules and join them together or assist and take in one big molecule, in this case, in this picture, and help crack the bonds out, holding it together so they're separated more easily. But it's generally a big protein that helps mush molecules together or snap molecules apart. All right, so features of all enzymes. They're proteins. So proteins, you probably know, are made of amino acids, and they end up folding up into big blobs. But generally speaking, when we draw it out, we just draw an enzyme as like a big old circle or square with a little dent inside of it. All enzymes have little separate places where the substrate or the reactant that it's going to act on fits into the enzyme, okay? So all enzymes are proteins that can allow reactions to go faster or use less energy. And oftentimes, if they need energy to help the enzyme to work, we're grabbing it from ATP. But anyways, they are specific to only one substrate, meaning that only one reactant and one reaction type can fit into the enzyme. That's why oftentimes when people draw out enzymes, they draw it out with a weird shape. And molecules, only specific molecules can work with a specific enzyme. So an enzyme that breaks down proteins cannot work on sugars. An enzyme that breaks down sugars cannot work on DNA. Okay, so they are very specific to whatever they're working on. And the word substrate refers to the reactant, the stuff that's going into the reaction and is going to be changed. They're also reusable. So when this enzyme works on a substrate, that substrate can be, when it is made into a product, it can then leave and that enzyme can be reused again. Okay, so just a heads up. This is a classic picture of sort of this yellow enzyme not working on molecules that don't fit inside of its special site. Okay? But if it does fit in its site, then that enzyme can be reused for a molecule over and over again. And once that molecule leaves, it can be working on a different molecule of that same type, right? So once this starch is broken down, it can work on another starch, and then another starch, and another starch. So they're all reusable. However, because enzymes have a very special shape, they're also really sensitive to the environment. So if the environment isn't ideal, that shape can be lost and it will stop working because stuff that needs to work on can no longer fit inside and be worked on. All right, so what is the factors that mess up enzymes? We're going to go into that. Okay? So the term referred to when our enzyme's shape gets lost so it can no longer work on the reaction is called denaturation. Okay, so our enzymes can either be permanently destroyed or their rate may change if the environmental conditions are not ideal. Meaning if homeostasis isn't perfect, our enzymes either slow down, and if the conditions get really extreme, they basically die. They basically get destroyed. Okay? So sort of like an egg that's fertilized can maybe turn into a chick if you heat it up just a teeny bit, but if you boil it, you basically have a cooked egg, a boiled egg. Okay? So in this case, denaturation is when the enzyme has been completely cooked or destroyed, so it can't work at all. Okay? However, if it's not under perfect condition, it may just not work as well or slow down. Sometimes it'll stop to the point where it won't work for now, but then it can recover. That is not called denaturation. That's called inhibit or inhibition. So if something gets inhibited or undergoes inhibition, what happens is the enzyme slows down or stops its work, but it can recover. So one classic way of inhibition is to use a molecule to block off the site. So this is a normal enzyme where the green reactant or substrate fits into the enzyme, and when it goes into the enzyme, it then gets worked on and produces product over here. However, see, I wrote inhibition here. I meant that instead of inhibit. An inhibitor molecule might block off the site so the reactant can't go inside, and in this case, my enzyme won't work at all. So inhibition can occur just by having slightly not great conditions to make it slow down or stop working, or it can actually be directly blocked off by a molecule. So a lot of drugs that maybe block pain, that maybe block acid production, like antacid, all of those a lot of the time work through inhibition by blocking off enzymes that would help otherwise create stuff that you don't want in your body. All right, activation is something that improves your enzyme's ability to do its job, that either allows the enzyme to work but it doesn't allow it in a temporary way or in a permanent way. So it's basically something that has to be added to make that enzyme work, but when it goes away, it'll stop working. All right. So in this case, for instance, this reactant that wants to use the enzyme doesn't fit until another molecule snaps inside of it, and then the machine has been assembled so this molecule could go inside and work. Okay? A lot of times the essential minerals and vitamins your body needs are activators, are things that help make that enzyme work when it otherwise wouldn't. So we should know denaturation, and then two things that do affect your enzyme function without permanently destroying it: inhibition and activation. Okay, so let's go over the things that can either denature or maybe inhibit or activate. All right, so body temperature, there's a big reason why we want to maintain homeostasis in it very carefully by sweating or shivering a lot. Now, if it's too cold, you can recover. It inhibits or slows down enzymes so they don't work as quickly. So in this case, being very cold can be dangerous because you may slow things down to the point where your body can't do things quickly enough to sustain life, but it's not as dangerous as being too hot. Okay? Being too hot very quickly can destroy your enzymes permanently. Okay? So fevers of 104 degrees, which is only like 6 degrees higher than your normal body temperature, that's enough to create death. So as soon as your temperature reaches 103, 104, you really ought to go to the hospital and get help. Okay? Now, pH is another one, acid base. So in this case, pH is the amount of H plus ion you have in your body. You may or may not have learned this in chemistry. Some chemistry classes covered it, some of them didn't. But in this case, you should probably know the idea of acid, and you know acid is sour, so acidic foods are sour. You know your stomach is acid. Well, in this case, acid has a pH of under 7, and what that basically means is you have a lot of extra H plus. Okay? Basic means you don't have as much H plus. You have very little of that, and typically things that are soapy or bitter are basic. So any cleaner, generally speaking, like bleach and stuff, most of those are basic. Okay? And then neutral is like water. It's just kind of not acidic, not soapy, nothing. So neutral. Okay? Now, the thing about enzymes are whatever tissue creates that enzyme wants to work at the pH of that tissue. So different pockets of your body, different organs in your body have different pHs. So if you have an enzyme that works in your stomach, for instance, it wants to have a pH of 2 or 4. It wants to work at an acidic pH. While if you have an enzyme in your intestine, it wants to work at more of a neutral range. And if that enzyme is placed anywhere else that is not in that range that it wants to work at, it will also stop working, i.e. it will denature at the wrong pH. So in this case, unlike temperature where it denatures at the high temperature, but not necessarily the lower one, with acid, if it's too high or too low, the pH, it will denature either way if it's at the wrong one. Okay? So our body needs to be very careful with pH. And what it does is it has buffers, special molecules that help keep the pH in the right range that will prevent you from dying. And that's another version of homeostasis. We'll learn about one kind, which is found in your blood. It's called bicarbonate, and it's really important for keeping your blood at the right level so your red blood cells can work and do its job of moving oxygen around your body. Okay, so we have pH, we have heat, and we talked about buffers. And yeah, blood pH is really important. Basically, 7-ish is the right amount. And if you even go to the 8-9 range or the 6 range, you're dead. Okay? So it's a lot less tolerable of acid changes. Okay. And like I mentioned, your blood has special buffers that will keep it at the right range. We're not going to go too into it, though. You may have learned about it if you took more advanced biology, but if we're going to learn it, it'll happen really towards the end of the year. Okay. Last but not least, we're going to talk about on-off switches. So there's special factors that temporarily change the function of your enzymes without denaturing them. So a coenzyme is an activator, and it works basically as a temporary on switch where its presence allows an enzyme to do its job to work better. You may need minerals or vitamins that work as these coenzymes. So you constantly need more vitamin K, vitamin B, vitamin C to constantly replenish these missing coenzymes. Otherwise, those enzymes won't work and parts of your body may not function as well as they ought to otherwise. So vitamin C, for instance, is needed for white blood cell formation. So that's why low vitamin C can be associated with lower immunity function. Though if you take in too much vitamin C, it's not going to make your immune the best ever. There's a limit to how much you can take in. But that's still the basic idea. Without these critical vitamins, you're not going to do certain things that your body needs to be done. An inhibitor works as a switch, as an off switch without destroying the enzyme. Okay? Well, why is that important? Well, if you destroy the enzyme, if you need to use that enzyme later, reuse it, you have to wait until your body remakes it, and that can take a while to get replenished. But if you only want to temporarily stop a reaction from occurring, but maybe be able to use it later, then you can use inhibitors. Okay? So in this case, my reaction substrates will go into my enzyme to do the reaction. But if it's overproducing, I can use an inhibitor to block it. My body can produce its own inhibitors, or sometimes if your body isn't doing homeostasis correctly, that's where drugs come in. So a lot of drugs actually function as the inhibitors that keep that enzyme from continually working when it shouldn't, when your body is overdoing things that shouldn't overdo. So for instance, I use Pepsid AC a lot because I have I overproduce acid, and that actually reduces the acid in my stomach because it blocks off an acid crater inside my stomach. Okay, so that's a classic example. Arsenic is a poison that's very dangerous because its job is to actually stop the creation of ATP. So the enzyme that makes ATP stops with the inhibitor. And, well, it only temporarily does it, but because your body needs ATP, by the time that inhibitor goes away, you probably ran out of ATP to keep your brain alive, so you're already dead by the time the inhibitor leaves. So arsenic is very, very dangerous as a poison. All right, so just kind of a sum up, the things that permanently denature your enzymes are the wrong pH and high temperatures, but they have to be kind of an extreme to get to that destroyed stage. If it's only slightly off, but not super bad, then it may slow down your enzymes, but until the shape literally alters, until it gets real extreme to the point that it cooks, then it's not denatured. But when it gets to that point, then it'll stop working forever. You can't recover. However, things that can change the speed without destroying your enzyme, and then you can recover and reuse those enzymes if needed, are low temperatures and inhibitors and slightly off conditions. And then coenzymes kind of work in the opposite way in that it activates rather than stops. So it's an on switch with your enzymes being usually off. You turn them on when you want them. Well, inhibitors are used for when your enzymes are always working and then you turn them off. So it's an off switch that only lasts for a little bit. But either way, these all can change whether your enzyme is working fast or not as fast without losing the enzyme itself. So, key takeaways. Enzymes are a type of protein that allows metabolic reactions to happen at a speed that can sustain life. So, nucleic acids are the ATP energy that may fall into helping to fuel your enzyme reactions, but the actual enzyme itself is a protein. All right? Enzymes are unique to their reaction, meaning each type of enzyme, each unique kind of enzyme works with only one type or one unique type of reaction. However, each of those enzymes can be reused over and over and over again, but they are sensitive to the environment. Homeostasis is all about maintaining conditions or environmental conditions for those enzymes to work as they should. But if conditions change in your body to change the environment, then those enzymes can potentially become denatured, meaning permanently damaged, and stop working, which can be very dangerous. Many drugs and nutrients alter enzyme function in a way to help either promote activity or reduce activity in response to whatever your body is doing. If your body is doing something incorrect, you can take drugs in a way to put your body back to normal if your body is unable to do homeostasis on its own. All right, that concludes the notes. Move on to any other work you have. Bye.